everyone, today I will talk about the procedures of our experiments and the steps of procedures. First of all, we prepare digestive medium and we will dissect subcutaneous adipose tissues from pigs. And the other step is collection of pigs' adipose-derived stem cells from stromal vascular fractions and identification stem cell surface markers of pigs' adipose-derived stem cell by flow cytometry. And the last step is differentiation of porcine adipose-derived stem cells into adipocyte, osteocyte, and chondrocyte. My friend will tell the two procedures of this experiment. Thank you.
Hello, in this experiment, subcutaneous adipose tissue from porcine is used in 60 gram. All steps are done at room temperature unless it is indicated. First step is preparation of digestion medium. Collagenase 2 powder with a total of 54,000 units is sold in 19 milliliter dimem in serological bottle to obtain 900 units per 1.5 milliliter dimem. Bottle is placed onto shaker for 15 minutes for agitation. Then it is passed through filter for sterilization. Medium is placed at 4 degree until usage. Second step is dissection of adipose tissue from pigs. All plastic and glass instruments are sterilized and medium is placed at room temperature. Piglets are sacrificed by an appropriate method such as electrical stoning or exsanguination accordance with a regulation. Dissection is done carefully and immediately. Piglet is laid on a surgical table in back facing up and belly down position. All hair of piglet from neck to tail is shaved off. Pig's dorsal skin is scrubbed with povidone iodine with 3 times, then iodine is left on surface for 10 minutes. Povidone iodine is removed by using 70% ethanol spray for several times. Gauze pads or tissue papers are used to clean visible povidone iodine on surface. Porcine dorsal fat layer of subcutaneous adipose tissue are separated along with the attached skin layer from muscles with the help of forceps. Then sample is immediately immersed into sterilized beaker containing serum-free dimem. Beaker is placed in a laminar flow cell culture hood after spraying outside of beaker with 70% ethanol. Hood is placed with large sterilized triple layer foil. Then tissue with skin is placed onto foil in facing down position. Remaining muscle cells are separated from adipose tissue by using forceps and scissors to avoid muscle contamination in sample. Fat is cut into squares, then pieces are placed in new sterilized beaker containing serum-free dimem. One piece of fat is placed onto customized slice holder with carbon steel slicer of skin layer on top and fat layer on bottom. Then tissue is sliced into 1 mm thick pieces, avoiding by slicing skin. Sliced pieces are minced with scissors as fine as possible. Minced pieces and filtered digestion medium are mixed in Erlenmeyer flask. Then flask is incubated and swirled in orbital shaker for 19 minutes for digestion at room temperature. It is checked for every 15 to 30 to prevent overdigestion. It should be slurry medium without tissue pieces. Equal volume with digestion medium, dimem 10% FPS is added to stop digestion. Thank you.
3. Collection of porcine adipose-derived stem cells from the stromal vascular fraction. The digestion medium containing the digested adipose tissues, though a single layer of chiffon, is passed into a clean sterilized
250 milliliter aluminum flakes or a 250 milliliter cellological bottle. A forecast is used to depress the middle of the chiffon to guide and assist passage of the digest. The chiffon is drained and twisted with a forecast to complete passage. The digestion medium is distributed into 450 milliliter conical centrifuge tube. These tubes are centrifuged at 700 xg for 10 minutes to collect the pellet of stromal vascular cells. The supernatant is decanted without disturbing the pellet to remove most of the top fat layer containing mature adipocytes. The pellet is washed by adding 10 milliliter DMAM into each tube to resuspend the pellet with pipetting and gentle shaking of the tube. These tubes are centrifuged at 700 xg for 6 minutes and the supernatant is decanted. 10 milliliter lysis buffer is added and then the pellet is suspended by pipetting. These samples are waiting for 7 minutes at room temperature to lyse red blood cells in the stromal vascular fraction. Equal amounts of DMAM, about 10 milliliter, is added to stop the reaction with a gentle shaking of the tube, and then these tubes are centrifuged at 700 xg for 10 minutes. This supernatant is decanted again and 10 milliliter DMAM is added into each tubes. The pellet is suspended with repeating pipetting and centrifuged at 700 xg for 6 minutes. It is repeated twice. The DMAM is collected and passed with suspended cell to a 100 micrometer strainer into a new 50 milliliter conical tube. The medium is pipetted gently several times to mix well, and 20 microliter of cell-containing medium mixed with 180 microliter of 0.4% terpombolus solution is liquid in a new 1.5 milliliter appender tube, and the cells are counted with a hemocytometer, and then Porcine Adipose-Derived stem cells is seeded at a density of 60,000 cells per centimeter square on the desired side of culture dish or plate with culture medium containing DMAM F12, 10% fetaborin serum and 1% antibiotics of penicillin, citromycin and amphotericin B solution. Generally, these stem cells is seeded on the 6-well tissue culture place for adipocytes or osteocyte differentiation, and the place or dish are incubated in the 37 centigrade degree incubator in air with 5% carbon dioxide to allow cell attachment to the plates. And fourth step is identification stem cell surface markers of Porcine Adipose-Derived stem cells by flow cytometry. After 24 hours, the medium is removed completely. The 10 centimeter dish is washed twice with PBS, and cells are harvested with 1 milliliter of 0.25% terpsion at 5 minutes at 37 centigrade degree. Trypsin is neutralized with equal amounts of culture medium, 1 milliliter about. Cells are collected in a new 15 milliliter conical tube and then are centrifuged at 400 xg for 7 minutes. This supernatant is decanted and the pellet is washed twice in 3 milliliter ice cold wash buffer using resuspension by pipetting cupped with centrifugation at 400 xg for 7 minutes. And these stem cells are suspended, counted, and adjusted to 10 times 6 cells per milliliter in ice called FCS wash buffer. cells were placed into multiple new 15 ml conical tubes, and tubes containing these stem cells are incubated at 4 centigrade for 30 minutes with antibodies against either phycoerythrin conjugated CD4A, CD29, CD31, CD44, CD45, CD90 for direct staining. The reaction is stopped by washing the cells twice in 10 ml FCS wash buffer cup with 400 XG centrifugation for 7 minutes. Cells are fixed and resuspended in fixation buffer for flow cytometry according to instruction and publication.
And I will talk about the differentiation of porcine deposited stem cell into adipocytes, osteocytes, and chondrocytes. First step is differentiation of porcine deposited stem cells into adipocytes. We first prepare adipose induction medium and adipose maintenance medium. For a adipose induction medium, 1 liter of serum-free DMAM-F12 is prepared with antibiotics of 1% of penicillin, streptomycin, and amphotericin B, containing the following: 1 ml insulin stock, 1 microliter T3 stock, 200 microliter transferrin stock, 100 microliter dexamethasone stock, and 100 microliter reciprotizine stock. Insulin stock is prepared by dissolving 10 mg per milliliter insulin in HEPAS buffer, which has pH 8, and transferrin stock is prepared by dissolving 50 mg per ml transferrin in double distilled water, and reciprotizone stock is prepared by dissolving the reciprotizone in 10 millimolar reciprotizone in DMSO. Adipose maintenance medium with the same addition as induction medium, but with omission of dexamethasone, is prepared. The second step is differentiation process of adipocyte. It should be noted that after seeding porcine adipose-derived stem cell on 6-well plates, porcine adipose-derived stem cell will be confluent within 72 hours. After 3 days, the medium component is removed, and then 3 ml of adipose induction medium is added into each well. The plates to incubator are returned. This is the 0 day of differentiation. After 3 days, the adipose induction medium is reviewed completely. 3 ml of adipose maintenance medium is replaced with every 3 days. Major adipocytes will be terminally differentiated by about 90 days after. Over 90% of adipocytes are well differentiated using this protocol. These adipocytes are ready for staining with red O state. And second step is differentiation of porcine adipose-derived stem cell into osteocytes. Osteocyte induction medium is prepared first, which is containing the DMAM-F12 media with 10% of fetal bovine serum and 1% of penicillin, streptomycin, and amphotericin B, containing 1 microliter dexamethasone, 1 millimolar beta-glycerophosphate, and 50 microgram per ml ascorbate 3 phosphate. It should be noted that after seeding porcine adipose-derived stem cells on 6-well plates, porcine adipose-derived stem cell will be confluent within, again, 72 hours. After 3 days, the medium is completely removed and 3 ml of osteocyte induction medium is added into each well. The plates are returned to 37 degrees incubator, and it's also the 0 day differentiation. Medium is replaced with osteocyte induction medium every 3 days. Mature osteocyte will form by 30 days of differentiation, 14 days of differentiation. These osteocytes are ready for alizarin red staining. Differentiation of porcine adipose-derived stem cells in chondrocyte is the other step. Chondrocyte induction medium is prepared by alpha DMAM containing 1% of fetal bovine serum, 6 mg per ml insulin, 50 mg per ml ascorbate 3 phosphate, and 10 nanogram per ml transform growth factor beta 1. Differentiation process of chondrocyte. After porcine adipose stem cell is seeded on 10 cm culture dishes for 24 hours, culture medium is removed completely and washed dishes twice with PBS. PBS is phosphate buffer saline. Porcine adipose stem cells is trypsinized with 1 ml of 0.25% trypsin ether for 5 minutes and then neutralized with 1 ml culture medium. Porcine adipose stem cells is collected in conical tubes which has volume 15 volume with a density of 2,500,000 cells per tube. Hemocytometry is used to count cells. After centrifugation at 400 G for 7 minutes, the spermaton is discharged without disturbing the pellet, and 1 ml of condensate induction medium is added in a 15 ml tube. The tube is returned to 37 incubator. It's also this is the zero of differentiation. The last step is condensate induction medium is replaced every 3 days without removing the cells on the bottom. Mature chondrocytes will form in approximately 14 days. The chondrocytes are ready for toluidine blue or staining now. And sixth step is staining of differentiated adipocyte osteocytes and chondrocytes. First of all is oil or staining for differentiated adipocytes. At day 9, adipocyte maintenance medium is removed in 6 well culture plates with differentiated adipocyte, and the plate is washed twice with PBS. Adipocytes are fixed with 10% formalin solution for at least 10 minutes. 10-minute formalin solution is removed and the plate is washed with twice double distilled water. After 2 washes, 100% propylene glycol is added to the culture plate and waited for 1 minute. 100% propylene glycol is removed and then oil-red O solution is added to the culture plate. Solution is waited for at least 10 minutes on the rocker shaker with gentle agitation at 100 RPM. The oil-red O staining is removed and then replaced with 60-100 propylene glycol. The solution is waited for 1 minute at room temperature. The 16% propylene glycol is removed and then the plate is washed twice with double distilled water. 10% formalin solution is replaced. The stained lipid droplets inside of adipocyte are studied for observation by light microscope. And the second step is alizarin red S staining for differentiated osteocytes. At day 14, osteocyte induction medium is removed from 6 well plates with differentiated osteocyte, and the plates are washed twice with phosphate buffer saline. Osteocytes are fixed in 10% formalin solution for at least 10 minutes. The 10 minutes formalin solution is removed from each well and the plate are washed twice with double distilled water. After 2 washes, 2% alizarin red S solution is added to the 6th well and waited for at least 15 minutes on a gentle worker shaker at 100 RPM. The alizarin red S solution is removed and then the plate is washed twice with double distilled water. 10% formalin solution is replaced. Osteocytes are ready for observation by light microscopy. And the last step is toluidine blue O staining for differentiation of adipose tissue in the chondrocytes. At the day 14, chondrocyte induction medium is aspirated from the 50 ml conical tube without removing sediments of differentiated chondrocyte in bottom of the tube, and the tube is washed twice with PBS. Chondrocytes are fixed in 10% formalin solution for at least 10 minutes, and the 10% formalin solution is removed from each tube and the tube is washed twice with double distilled water. After 2 washes, pellet is embedded in OCT compound and sectioned with cryostat at a thickness of 5 micrometer. Slide of cryostat selections are stained with toluidine blue O solution. Toluidine blue O solution is removed and the section is washed twice with double distilled water. Chondrocytes on slides are ready for observation by light microscopy. So we differentiated these tissues into 3 types of tissues, and then we will stain these tissues by using different staining techniques. Thank you.